Welcome to an in-depth exploration of enzymes, the remarkable molecules that drive life's essential processes. Enzymes are biological catalysts, proteins that accelerate biochemical reactions without being consumed in the process. They are crucial for nearly every metabolic function, from digestion to DNA replication. Their efficiency is astounding, speeding up reactions by factors of millions or even billions. This incredible power comes from their high specificity, meaning each enzyme typically acts on only one or a few specific substrates. Today, we'll delve into their classification, how they carry out their activity, and the intricate mechanisms of enzymatic reactions. Enzymes are systematically classified into six main categories by the International Union of Biochemistry and Molecular Biology. First, we have oxidoreductases, which catalyze oxidation-reduction reactions. These enzymes are vital for processes like cellular respiration, where energy is extracted from nutrients. Next are transferases, responsible for transferring functional groups from one molecule to another. Think of kinases, which transfer phosphate groups, playing a key role in energy transfer and signaling pathways. Hydrolases catalyze the hydrolysis of bonds, essentially breaking them apart using water. Digestive enzymes like amylase and lipase are prime examples, breaking down complex food molecules. Lyases catalyze the breaking of various chemical bonds without hydrolysis or oxidation. They often form new double bonds or rings, playing roles in synthesis and degradation pathways. Isomerases catalyze the rearrangement of atoms within a molecule, converting one isomer to another. These are crucial for maintaining metabolic balance and preparing molecules for subsequent reactions. Finally, ligases catalyze the joining of two large molecules by forming a new chemical bond. This process typically requires energy, often supplied by the hydrolysis of ATP. DNA ligase, for instance, is essential for repairing DNA strands and in DNA replication. Now, let's explore how enzymes actually perform their work, focusing on their activity. Central to enzyme function is the active site, a specific region on the enzyme where the substrate binds. This site is uniquely shaped to accommodate its specific substrate, much like a lock and key. The lock and key model suggests a rigid fit, where the enzyme's active site is preformed to match the substrate. However, a more refined understanding is the induced fit model. Here, the enzyme's active site undergoes a conformational change upon substrate binding, optimizing the fit. This dynamic interaction enhances catalytic efficiency by bringing reactive groups into optimal alignment. Several factors significantly influence enzyme activity, starting with temperature. Each enzyme has an optimal temperature at which it functions most efficiently. Beyond this optimum, higher temperatures can cause denaturation, where the enzyme loses its 3D structure and function. Similarly, pH plays a critical role. Enzymes have an optimal pH range. Extreme pH values can alter the enzyme's charge distribution, disrupting its active site and leading to denaturation. Substrate concentration also affects reaction rate. Initially, increasing substrate leads to faster reactions. However, at high concentrations, the enzyme becomes saturated and the reaction rate plateaus. Enzyme concentration directly correlates with reaction rate. More enzymes mean more active sites available. This allows for a faster conversion of substrate into product, assuming sufficient substrate is present. Finally, inhibitors can regulate enzyme activity, either reducing or completely blocking it. Competitive inhibitors mimic the substrate and bind directly to the active site, preventing the actual substrate from binding. Non-competitive inhibitors bind to a different site on the enzyme, altering its shape and reducing its activity. Understanding these factors is crucial for comprehending how enzymes are regulated within living systems. Let's now dive into the detailed mechanisms of enzymatic reactions, step by step. The first step is enzyme-substrate complex formation, where the substrate binds to the active site. This binding is often reversible and involves weak interactions like hydrogen bonds and van der Waals forces. Once bound, the enzyme facilitates the reaction by stabilizing the transition state. Enzymes lower the activation energy required for the reaction to proceed, significantly speeding it up. This stabilization is key to their catalytic power, making reactions feasible at physiological temperatures. After the reaction, the products are released from the active site. Crucially, the enzyme itself remains unchanged and is ready to bind to another substrate molecule. This regeneration allows a single enzyme molecule to catalyze thousands of reactions per second. Some enzymes require assistance from cofactors or coenzymes to function properly. Cofactors are inorganic ions, while coenzymes are organic molecules, often derived from vitamins. They assist in catalysis by carrying electrons, atoms, or functional groups, or by stabilizing the enzyme's structure. Without these helpers, many enzymes would be inactive, highlighting their critical role in metabolism. In summary, enzymes are indispensable biological catalysts, orchestrating the myriad reactions essential for life. We've explored their diverse classification, from oxidoreductases to ligases, each with a unique function. We've also seen how their activity is governed by the active site, the lock and key, and induced fit models, and how factors like temperature, pH, and concentration finely tune their performance. Finally, we dissected the reaction mechanisms from substrate binding to product release and the role of cofactors. The complexity and elegance of enzyme function truly underscore their fundamental importance in all living organisms.